This is where the concept of epigenetics comes in. It's the bridge between genetics and the environment. It's how factors out in the environment determine what genes are turned on and off. Okay, so for example, let's say you were in a car accident and got injured. That's a signal from the external world that enters your body through different chemical reactions, hormones, molecules, molecular structures. And these factors get into our cells and they start interacting with DNA, okay? And as a result, you start sort of decorating your DNA with different groups. And that tells a particular gene to turn on or off. Okay, so let me zoom back out. Something happened out in the environment. A signal was sent into your cell. Different molecules were added to the DNA and certain genes were turned on and off. Okay, and depending on what that signal was, you're going to turn on different genes. If it was something bad, you're going to maybe turn on the inflammatory genes. If that thing was good, like a good night's sleep, you're going to turn on the good genes. So all of the things that happen out in the environment are just information that gets into the body. And epigenetics has really broad effects on all body systems. So it's going to impact your immune and nervous system. It's going to impact how your mitochondria work. It's going to influence oxidative balance. Remember, I said too many unstable molecules is not good. So when you get exposed to a lot of toxins, like let's say BPA from plastics, that can actually put a stress on the systems in your body, and then you get that oxidative stress. So epigenetics is highly sensitive to the environment. It's constantly looking to see what's happening in the environment so that you can determine what you're going to do next on a genetic, cellular, biochemical level. And these changes that are epigenetic, they're reversible, right? You can turn things on and then you can turn things off. You can eat a hamburger and then you can eat, a plant-based meal, and you're just going to keep turning on different sets of genes. So just to give you a quick idea of what that looks like, here's methylation. This is DNA, and these little black popsicles are chemical groups. They're methyl groups added to DNA to create attention to say, hey, turn on this gene, right? Methylation here may have happened based on something that happened in the environment. Another type of epigenetic change is histone acetylation, which is basically putting these chemical groups onto DNA to unravel DNA so that now it's open and exposed and different machinery can come in and start reading the genes. I mean, it's just really fascinating, right? And all of this happens through chemical structures. Okay, we're going to actually zoom in onto some very microscopic pieces of your body that you've probably never thought about. This is a very complex topic, but I just want you to kind of have this visual of this system existing in your body. PPAR gamma is a system of DNA receptors. They bind proteins and they turn genes on and off. And typically what PPAR gamma turns on is the good stuff. And I always think of the G in gamma as good. So PPAR gamma is a good guy. And when the system is turned on through epigenetics, remember you decorated the DNA and the PPAR gamma turned on and it started reading genes that turned on anti-inflammatory genes. We also know that it's neuroprotective. So it's good for the brain. It's good for that neurodegeneration that can happen with a condition like MS. And I put a picture here of greater burdock. You may see this like growing on the edges of sidewalks. It's a root with some green leaves, and it contains arctogenin, which has been shown actually to reduce inflammation and demyelination in mice that are used to study MS. And we think it works through PPAR gamma. So what do you have to do? If you were to eat greater burdock, you would turn on your PPAR gamma system, then you would turn down inflammation and you'd protect the brain, okay? And by the way, EAE mice, this is a mouse model of MS, and And this is, what does it stand for? Experimental autoimmune encephalomyelitis. Basically, they take mice, they inject them with myelin basic protein, which is a protein that the mice can start making antibodies to, and then they end up using those antibodies to attack myelin. So effectively, you're giving them MS. And then you put them through experiments to see, what the effect of whatever you're studying is. Okay, so the point of this slide was to get you to think about PPAR gamma. That's a good system that works through epigenetics. Here's another system, NF-kappa B. I think of the B as bad. This much like PPAR gamma is a nuclear transcription factor. It's involved in reading different genes, and its effect is basically to turn on inflammation. And we know that it plays a role in many things, like in neurodegenerative conditions, autoimmunity, cardiovascular disease, cancer. So you basically want to keep NF-kappa B quiet.